Our first speaker in this section is Jeanne Loring. Jeanne is a Professor Emeritus at Scripps Research Institute, founder and founding CSO at Aspen Neuroscience, board of director of Summit for Stem Cell Foundation. She is the author of more than 250 publications, inventor of PluriTest. Jeanne developed one of the first NIH-funded human embryonic stem cell courses, so she has another well-known title as the godmother of stem cells. Many young scientists, including our employees at iPSCs, are familiar with her book, Human Stem Cell Manual and Laboratory Guide, which is considered the Bible for stem cell culture. So her work is still continuing to inspire a lot of young scientists and entrepreneurs here.
Yeah, thank you very much. There's a new edition of this in the works, just in case you're wondering, and I may be asking some of you to write chapters for it because I just got the first of the year reminder that I have an obligation to finish this book. Okay, so I'm going to tell you two stories today. I'm going to tell you a story about how we went from stem cells to a therapy for Parkinson's disease, which is going to start, I hope, within the first quarter of this year. And I'm also going to tell you a very short story about other work I'm doing right now, which involves sending neural organoids derived from iPS cells to the International Space Station to try to find out what the effects of microgravity to perturb them in a different way. Whatever reason, it's a lot of fun. Okay, so I have disclosures. I founded Aspen, and I said there's a clinical trial. I am scientific advisor for a number of companies, in fact, more than this now. Novoglia Conception, Symbiosis, and CademoStem, and I'm also an ethics advisor for Merck in Germany. And I've done a lot of bioinformatics and I do machine learning, but I'm really interested in AI. And my main use of AI right now, ChatGPT, is to ask it to write haikus. So this is my haiku to begin. Unlocking life's code, 25 years of promise, stem cells light the path. And it's been 25 years. I've worked on these cells for 25 years. As you know, pluripotent stem cells are unique. They aren't like other stem cells that you find in your body. They exist only in cell culture. It's important to note that they're the only non-cancer cells that don't senesce, that continue to divide in culture. And they're also able to differentiate in any cell type in the body. And as you know, there are two sources. One was the the first human embryonic stem cells were reported in 1998. The first mouse embryonic stem cells were reported in 1981. So we're really standing on the shoulders of giants here. This has been going on for a very long time. We just happen to be working on humans. There are a number of pluripotent stem cell therapies that are now in clinical development, including age-related macular degeneration, Parkinson's disease, epilepsy, lipodystrophies. These are using different cell types that are derived from iPS cells. And in most cases, it's a cell replacement therapy, but not always. There is a company here in San Diego that's working on hair loss using epidermal cells from iPS cells. iPS cells are being used for cancers by Fate and Shoreline here in San Diego. Inflammation is being treated with mesenchymal stem cells, which are the cells that you would find in fat, for example, of us right now. But in this case, they're making them from pluripotent stem cells, from iPS cells. And that, as you know, has all the advantages of being able to get unlimited quantities of cells and to be able to characterize them really well before you use them in the clinic. Heart disorders have been really challenging. Blue Rock, HeartWorks, and StemCardia. StemCardia is a new company started by a group at University of Washington, Chuck Murray, and also type 1 diabetes. And there are a couple of companies working on this. There's actually a lot more than I have listed here, Vertex and Sana. These are kind of the easy targets right now. And so there are multiple groups working on each one of these. What did I do with my thing? Here it is, I got it. No rescue necessary yet. So Parkinson's disease is caused by the loss of a particular type of neuron in a particular part of the brain. The part of the brain is called the substantia nigra, and the symptoms don't arise until about half of those cells have died. It's progressive, so those cells will continue to die and the disease will get worse. There's a drug therapy that allows people to have a normal life for a fairly long period of time, but it doesn't help the loss of neurons. So there is no therapy that prevents the continued degeneration of dopamine neurons in this disease. But Parkinson's disease is especially good an early idea of it as a candidate for cell replacement therapy. And this goes back for another 30 to 40 years. There were a group of people who were treating Parkinson's disease patients with fetal tissue. This is before there were any embryonic stem cells or iPS cells. So they would take the precursors from an embryo, very small embryo, 6 weeks or so old, 6 to 10 weeks, and they take this little tiny piece out and then implant it into the brain in the part of the brain that receives the dopamine neuron input. And that was really important because that's a pretty large target to do a transplant, whereas to try to put the cells right back where they originated would require a smaller target. It is a smaller target, but also would require them to then grow processes back up to the striatum. So they put them directly into the striatum so they could connect really quickly. Now, this didn't work very often. It worked very seldom, but this is where hope comes from. Once in a while, it worked beautifully. There were a few patients who never had to take drugs again and were completely cured of the disease. So our goal was to make this more common, to make it possible to be consistent in treating the disease and reversing the symptoms. I wanted to point out Kurt Fried here, who wrote a book called Healing the Brain. It's his own personal story about the fetal cell transplants. It's a really interesting book. So in around 2017, there was a meeting in Kyoto. It was a cooperative decision for multiple groups who are working on cell therapies for Parkinson's disease to all get together and share their methods. And we published a paper about it. So this is really important. Having colleagues who are willing to share information with you over and over and over again is the only way that you can make one of these things work. None of us can do this on our own. The 4 groups are still the 4 groups that are starting clinical trials now. There was the group in UK and Sweden, Roger Barker and Malin Parmar. They were using embryonic stem cells, and similarly, a group in New York, this is Blue Rock, is using embryonic stem cells. Jun Takahashi in Kyoto is using iPS cells, but they're not matched to the patient. So all 3 of those require immunosuppression for at least a year. So this is being treated like a kidney transplant or an organ transplant. You have to be completely immunosuppressed. And we don't know whether you can actually ever come off immunosuppression because there's no information, there's no history of doing this in humans. So when we started doing this project in about 2012, this is the autologous iPS cell project, we decided to take advantage of the unique benefits of iPS cells, and that is personalized therapy. We can make those cells from any person. So all we had to do was to turn those into dopamine neurons and then get them approved for a clinical trial. And that took us a bit longer. This is a story that you could read in the Wall Street Journal. This is about our efforts, and it gives a really good overview of how iPS cells are generated and how they're used to treat Parkinson's disease. This really is about the company that I started called Aspen Neuroscience. So why would we use autologous patient-derived cells instead of a single line? It is harder in some ways. It's easier in other ways. So I think it really evens out in the long term. If you're using allogeneic, which is unmatched, that means that you do have to immunosuppress the patients, no matter what the therapy is. Manufacturing is in the form of scale up, and so you make hundreds or billions of cells in a big bioreactor. The problem starts coming when you start worrying about something that happens over and over again, iPS cell lines, and that's genomic instability. When cells divide, remember I told you, they're just like cancer cells, they can proliferate forever. Well, they're just like cancer cells. Every time they divide, they acquire mutations. So that's something we always have to keep in mind. It's sort of the dark side of pluripotent stem cells. The last one is redosing, and there's a positive aspect of that. If the cells for some reason don't survive in the first transplant and we are given permission to do so, then we could transplant the cells again because there'd be no immune response, no buildup of immune response. So I want to talk a little bit about the perils because this isn't mentioned very often. And this is from a paper that we published quite a long time ago, 2011. And in this paper, we took 186 pluripotent stem cell samples from all over the world. This was more of a task of me showing up at conferences and asking people for their cell lines. That turns out, I think other people have used that sort of idea. You show up, you ask the researchers directly for stuff, and they'll give it to you. So that's how we gathered them all. And we analyzed them by SNP genotyping, which is the same methods used for 23andMe and Ancestry.com. It gives you an overview of the entire genome. So we found that there were a lot of copy number variations. You can see just in this diagram on the right side of the slide that everything that's red shows an example of a duplication. Sometimes the duplication is an entire chromosome. And what I'm trying to say is that if you don't look, you won't know, because none of these people who sent me their cells knew that they were abnormal. They all forgave me because they were co-authors on the paper. But a lot of them had to throw those cell lines away. But at least we knew that they were bad and they didn't invest any more time in them. So the reason why they acquire mutations is they have a what I call evolution in an incubator, and it really is evolution. A cell, one cell in a group of cells will have an error. That error gives it a selective advantage, just like selection, evolution. And then as those cells expand, that cell will proliferate. It'll be more resistant to stress. For whatever reason, it becomes dominant. So the whole culture has now evolved into a mutant culture. And those mutants are often associated with cancers. One of the ones that we see a lot is a lack or a mutation in P53. And we have found in 2017, in fact, P53 mutations were discovered in a whole bunch of embryonic stem cell banks. These banks were, in some cases, actually clinical grade cells. So you have a clinical grade bank, you've invested, I don't know, $1 million in it. You discover there's a P53 mutation in that bank, and you have to throw it away. Unless you can figure out some reason that the FDA will allow you to use cells that have a P53 mutation, because they are cancerous by definition. So what we did when we decided to do the autologous therapy is develop a lot of genomic methods in order to quality control the cells, because we had to make the cells each time from each individual. And in order to do that, we needed a lot of help in qualifying those cells to make sure that we got the same product at the end. So essentially what we did was this little loop here, you see, we start with somatic cells. We start with fibroblasts. That's more traditional than anything else. They're fine, but you can use other cell types. The somatic cells are analyzed by whole genome sequencing, and then again by SNP genotyping to look for structural changes. Then they're turned into iPS cells. We use a Sendai virus with the Yamanaka factors because the FDA thinks that's just fine, but it also works really well. And then at this stage, we have, instead of using like a panel of antibodies or some other kind of single marker analysis, we decided that we would look at the entire genome of our cells that were our iPS cells and our cells that were going to be used for transplants. So this is done by RNA sequencing. We developed two tools. One is called PluriTest, and that's widely used all over the world for making sure your cells are pluripotent when you start with that. But we also developed another tool called Neurotest, which is to make sure that the cells that we are going to transplant to patients are at the ideal stage. And this requires gene expression profiling and bioinformatics, but essentially it's not very hard, because what you need is a whole bunch of examples of things that work, examples of things that don't work, and then you can have your computer solve that equation and be able to predict what sort of profile works and which one doesn't work. So the whole genome sequencing is done actually at three times during this process. This is essentially what we presented to the FDA. So this is what Aspen is doing, whole genome sequencing 3 times, because it's really important not to have any dangerous mutations in these cells. And then the RNA sequencing is done twice. Oops, sorry. Okay, now this should show you. Let me see if I have to poke this. Yeah, here you go. You're going to come and rescue me? You're going to do it. You do it. Okay, so this is the assay that we use to show that the cells are efficacious. It's a rat model. What we do with these rats is to get rid of the dopamine neurons on one side of the brain. That results in them rotating. When you give them amphetamine, they'll rotate in the direction of that lesion. And so you can measure the number of rotations. That's how you quantify it. It isn't exactly Parkinson's disease, but it is really, it is definitely the same neurons. So this is the work that we used to present to the FDA. We showed that the rats that were lesioned and then treated with a transplant of human iPS cell-derived dopamine neuron precursors were recovered completely over time. So you can see that in those 2 colored lines, the blue and the green, that they return to normal over a period of about 24 weeks. Not fast, but the reason it's not faster is because they actually have to make synapses. These are young neurons. They have to make synapses in the brain in order to function. Oh, there's another one moving, and now another one moved. There you go. See, there's the transplanted one, not circling. Okay, so I'm going to show you briefly how our rationale for predicting the fate of cells. These cells can make everything, right? So how do we, when we differentiate them, how can we be sure that they are going to make the cell type that we need? Especially that is difficult when the cell type that you need is not going to be developed in culture, but is going to differentiate in the brain. We can't transplant fully mature neurons, they die. And it turns out, I'm going to show you in a moment, that we have to make sure that the cells are at exactly the right stage. And I'll show you how we did that. This is an example from PluriTest, and this has become a very popular tool for demonstrating that your cells are pluripotent. It's popular because it's free, at least it was free. I think it's still free. It's free, and it takes no work except for an RNA sequencing of your sample. You upload it, it comes back down, it tells you whether it passed or didn't pass. You don't have to know anything other than that. And that's why it was really popular. Second reason it was so popular is that journals accepted this as evidence of pluripotency. So we use the same model essentially to create Neurotest. In this case, we're trying to predict that cells that are at a neural precursor stage are going to become dopamine neurons. And we use exactly the same strategy. You just have to generate lots and lots of cultures and have some of them work and some of them not work. So that's what we have done. And we measured either the in vivo recovery of these cells, like I showed you earlier, or we showed that these cells become fully mature dopamine neurons. And that requires showing that they will release dopamine just like a neuron is supposed to in culture. They also fire a very typical bursting behavior if you do electrophysiology on them. So they're very easily identifiable once they're mature. but we needed to get a gene expression signature that told us that they would get like that, right? Okay, so this is a publication on the primary work that we did that outlined the entire thing that we submitted to the FDA. And that was our work to determine exactly when in culture, and when I say when, I'm talking about gene expression signature, not days. Gene expression signature changes over time, so where's the sweet spot in that? And we also were interested in discovering why some cultures worked and some didn't, in order to narrow it down. So what we discovered was that if we grew our cells, in this case for 18 days, under differentiation conditions, they would get recovery in the rats. If we grew them for another week, 25 days, which actually was the time point that was originally used in the rat studies long ago, they didn't work. However, they engrafted in both cases. So what's going on here? Well, you can see here on the left that there are effective cells and there are ineffective cells. In both cases, you see that the brown is tyrosine hydroxylase. That indicates where the dopamine neurons are. And you can see that there are dopamine neurons in both of those transplants, but only in the upper one were they effective. And the reason for that is that they grew out, they had outgrowth. They sent out neurites. They connected with the host cells. And you can see when we did the gene expression analysis, we discovered that a lot of the genes that are suppressed by a particular transcriptional repressor called REST were all lower in the cells that worked and higher in the cells that didn't work. In other words, there was a switch, and we had to be before that switch, because then a whole plethora of genes associated with neuron differentiation and maturation couldn't be on, or the cells wouldn't work. So even harder. We don't even, we can't just look at them and say they're going to be neurons. They don't look anything like neurons. Okay, so that study, and you can look just at the bottom of this, was approved by the FDA on August 8th last year. Aspen Neuroscience, the company that I started, got approval for a phase 1. It's actually a phase 1/2A. And the other three people that I talked about who were all in Kyoto are also all doing their clinical trials. One of them is Blue Rock, Sloan Kettering and Blue Rock. And they got FDA approval before the rest of us. And they have finished their phase 1 trial with 12 Parkinson's disease patients after 1 year. They reported on the 1-year results, which are not terrible, but not very impressive. That's the point at which they get rid of immunosuppression. So we're all looking forward to the future to see whether those patients get better or they get worse, because that's going to be a really important thing to know. So the other ones are Kyoto. Kyoto has taken a very long time, and they've been very quiet about telling us what's going on. I did learn from the PI that the cells are surviving in the patients, but they are not giving us any information about how they're doing. The Lund University, that's the Swedish and UK group, is now partnering with Novo Nordisk in a phase 1, which they started last November. So they're slightly ahead of us, but not by much, and we're still the only ones who are using autologous cells. So we don't have to worry about that immunosuppression, like when you take it off and when you put it on. I want to acknowledge this because this is really important. This actually evolved very much like some of the stories you've already heard about rare diseases. I couldn't get funding to save my life in 2011 to do this. I mean, it was people just said, I mean, they actually kept saying it was impossible until like August 7th of last year. And when we got FDA. So the way it started was a group of Parkinson's disease patients and their advocates climbed Mount Kilimanjaro, which is not a small mountain. It's 19,340 feet. And they had gotten a promise, if they did this, that they could raise funds to start development of this autologous therapy in my lab at Scripps Research. And then they did some other things. They went to Everest Base Camp. They did other hikes. And all in all, the Summit for Stem Cell Foundation, which was the group that does this, raised $3 million for me over this period of time. When we got the first work done, we started getting grants, which helped a lot. And then in 2018, I decided to start Aspen Neuroscience, and we have raised about $220 million to date. And that may sound like a lot of money. Anybody think that's a lot of money? It's a lot of money. It'll get through the phase 1 easily, and presumably get to the stage where the company can raise more money for phase 2. So we also got an FDA fast track designation, which allows more interaction with the FDA. These are the people I wanted to give credit to. When I started the lab, I actually left Scripps and I took my entire lab, all the people in my lab, all the equipment in my lab, all my grants, and I started Aspen. So I actually moved out from academia into biotech completely with the lab intact. And that turned out to be the best possible idea, because I had all these people at this company, the startup company, who had experience on this very project. And I also got credibility because we had grants that I could transfer over there. I also got to move all the equipment, which was extremely cool because it cost $1 million. And so we just essentially, it took us one day to go from being an academic lab to being a biotech company. All we did was keep going. So that was really important. And then we've also had support from CERM, from NIH, from the Silverstein Foundation, and from Aspen Neuroscience. So here's my haiku for the future of stem cell therapy for Parkinson's disease. Neurons bloom anew. FDA's blessing ignites hope. Parkinson's may fade. That's what ChatGPT thinks. Now I'm going to tell you this short story of what I've been doing over about the last 5 years, but now it's getting more intense. And this is to study the effect of microgravity on neurons. Now you can imagine, if you are an astronaut, they can take blood samples, they can measure your muscle strength, they can look at your bones and see how strong they are, but they cannot get into your brain. So in order to find out what happens to neurons on a space station, and presumably farther out in space, we were asked to start this project, which has turned out to be absolutely delightful. We've now had 5 missions, and it is funded through the National Stem Cell Foundation, which is the blue logo there. It's a nonprofit, and New York Stem Cell Foundation is our collaborators. Space Tango and Axiom Space have helped us get the cells up into space. So here's the haiku. Cosmic voyage starts, stem cells defy gravity, life blooms in space, space's embrace. Okay, so cells can make everything. So I just wanted to do a spin on this particular cartoon, which is very popular. You can be anything when you want to be when you grow up, and then I have the cell saying, I want to be an astronaut. So our project was to understand the effect of microgravity on the brain and focusing on neurodegenerative disease. This is the way we designed it. So there are a lot of different kinds of neurons in the brain. I think you know that. Only certain ones seem to be affected by certain diseases. So we chose, just because this is where our expertise was, just to study Parkinson's disease and multiple sclerosis, different parts of the brain. So for multiple sclerosis, we made iPS cells from people who had multiple sclerosis and a control. And we generated cortical neurons from them. For Parkinson's disease, we did exactly the same procedure that we used to transplant to patients. We had a Parkinson's disease patient, and we generated dopamine neurons. And this is where the really cool thing came about. We wanted to reproduce a part of the brain. In order to have the brain be have the organoid be sort of brain-like, it needed to have a key component of the brain, which is the brain's immune system. Because neuroinflammation underlies a lot of neurodevelopmental and neurodegenerative diseases. So we made two separate populations. We made the neurons and we made microglia. We had to do that because microglia come from an entirely different part of the body during embryonic development. They come from a structure called the yolk sac, and they migrate into the developing brain. The neurons develop in the brain, so we couldn't get them both in the same thing. So then we mixed them together to make organoids. And then the secret to our success was that we decided, since we were going to send them up into space, that we should, we wanted to design a sealed system. So what we did was, and everybody, this is another thing that nobody thought would work. This is really fun when stuff works. We took each organoid, put it into a cryovial, 1 ml cryovial. Those are sturdy little tubes. We put 1 ml of culture medium in it, sealed it up, set it up space for a month. And I'll show you what happened when we came back. So it takes a long time to get to space. I just want you to appreciate this is not a lab experiment. We prepare the samples, and then we have to ship them to the Kennedy Space Center. They get loaded into a device that is approved for putting on the space shuttle. And then there's a launch, and then it docks. All this takes time. And then it gets installed on the ISS, and then a month later it splashes down and it goes back to the lab and we analyze them. Okay. We've gotten pretty good at this over time. But this is the most important result, I think. First of all, that even though the cells were in those little vials, they came back not only alive, but they came back even healthier than our controls that were on the ground. And one of the things that we discovered from gene expression profiling is that the cells are more mature when they're in space than they are on the ground. Now, go ahead and hypothesize. We're testing my hypothesis right now, so I'm not going to tell you what it is until I find out whether I'm right. But there was less cell division and there was more maturity, which is, a really interesting and very clear-cut result. It didn't matter which cells they were. Both types of neurons do the same thing. So I want to acknowledge the help of these people. This is the group from we call our space team. One of the advantages of this, there are two advantages I'll mention to you. One of them you can see in the lower right, and that is I get to watch the rockets take off from the same place the astronauts' families get to watch. And then the other is that I get to send my own cells up into space. So if you see in the middle there, those little organoids, which you can barely see, those are from my own iPS cells. So we made dopamine neurons from my cells. I'm the control, which is, kind of a cool thing to do and worth 25 years of work on iPS cells. This is from our latest flight, which is an Axiom mission. And this was a private astronaut mission. There were international group. They're going to come down on Sunday. They've come away from, they've undocked from the space station, and now they're doing this orbit, which brings them lower and lower to Earth. And then they'll splash down on Sunday morning. Okay, I'm going to close with my stem cell haikus for the future of stem cells. Stem cells hold the key to healing and longevity. A bright future lies ahead. Thank you.